Bismillahirrahmanirrahim. We have another new video on coordination as requested by some of you. There's these questions on the eye which I want to just revise with you all so that you can understand it a little bit better. Now let's look at the first question. Question 1a. Figure 2.1 shows the reflex action that involves the eye. A shows an eye in dim light. B shows the same eye when a bright blue light is shone into it. Identify the stimulus to which the eye responds. Receptor cells that detect the stimulus, effector, and response that the eye makes. Write your answers in table 2.1. Now, what is a stimulus? Basically, it's light intensity. You could have said light, you could have said light intensity. Then receptor cells would be the rods and cones in the retina. Effector would be the circular muscles of the iris. And the response will be that the pupil gets smaller, because if there's too much light entering the eye, now that can damage the retina. Please do not say damage the eye. It doesn't damage the eye. The eye itself is correct, but what it damages is the retina, a part of the eye, not the whole eye, not the cornea, not the conjunctiva, not the lens. So be very specific when you are writing the answers to this. So light intensity, cones in the retina. Please remember that cones is not in the 5090 syllabus, but if you had written retina, that was fine. Circular muscles of the iris would be the effector, and the response would be the pupil constricts. Now here's another diagram which I want you to have a look at and revise why in bright light what happens. So the pupil becomes smaller. So circular muscles will contract in the iris and radial muscles will relax. While in dim light, what is going to happen is that the circular muscles will relax. So the pupil becomes a very large circle. This is the opening to allow more light to pass in. So the radial muscles will contract and the circular muscles will relax. That's why it becomes a bigger circle. Here you can see the iris is a smaller circle. So you can see the iris here on this side. And here you can see the iris here. So please be careful when you're talking about this. You must be very clear on this concept. And there is of course the diagram here as well, which you can see the actual photographs of the eye. Then part 2 of the question says describe how the nervous system coordinates the response shown in figure 2.1, the previous diagram that we just saw. So this is the one that we're talking about, figure 2.1. And now they're asking us how the nervous system coordinates this response. So basically first we have to talk about electrical impulses. So the light intensity is detected by the receptors or the sensory cells in the retina. Then this impulse travels across the sensory neuron. This will get you 2 marks because there's 1 mark for the electrical impulse and 1 mark is for the sensory neuron. Where is it going to go? It's going to go to the brain. You see, the eyes are just in front of the brain is at the back. Goes to the brain along the motor neuron. How the nervous system coordinates the response? So it goes to the brain, and then how is it going to come? From there, it is going to come through the motor neuron to the effector. The effector is the muscles of the iris. So initiated by the receptor cells in the retina along the sensory neuron or the optic nerve, electrical impulses will travel to the brain, or you could have said the central nervous system, and from the brain through the motor neuron to the effector. The effector in this case was the circular muscles of the iris. Question 2 is something to do a little bit with eye and a little bit on the Oxen story as well. Ahmed entered a very dark room. His irises responded by changing the pupil size, and gradually he could see shapes of objects in the room. Dust in the air made him sneeze. Suddenly the door slammed shut, causing the heartbeat to speed up. He switched on the light to find the door, and he grabbed the door handles. Complete the table by stating 2 voluntary actions and 2 involuntary actions described in the text above. This is a little bit on the 0610, so don't get upset if you are the 5090 ones. So what was the voluntary? Going into the room, switching on the light, grabbing the door handle. What was the involuntary? Pupils changed size, heartbeat speeded up, sneezing. So going into room, switching on light, grabbing the door handle. Involuntary will be pupils change size, heartbeat speeded up, and referred to sneezing. So there's more the IGCSE 0610 syllabus, which has these sort of questions. Now it says actions are caused by the stimulation of effectors. Name the two different types of effectors in the body. There are only two different types of effectors, muscles and glands, nothing else. So muscle 1 mark, gland 1 mark. Because motor neurons only supply muscles and glands. That's the only two things which responds to different stimuli. State the type of neuron that stimulates effectors. Motor neuron. So anything that can stimulate a muscle to contract or relax, and glands to produce secretions, like you feel like eating something, you hear this very you smell this very nice aroma of biryani being cooked, or chicken tikkas being cooked, or barbecue being cooked, so you start to salivate. So why salivate is because more saliva is going to be made by the salivary glands in anticipation of you going to start to eat, which will be helpful, of course. So the type of neuron that stimulates effectors are motor neuron, and the effectors are only two types. Either they are muscles or they are glands. Bas, that's it. No other story. Plants respond to stimuli such as light. State the name of the response of plants to light. Phototropism. Phototropism. You got 1 mark for that. You're not going to talk about the positive and negative story because that's not asked. They just said name. Ahmed was provided with several young plant shoots and a sample of auxin. Describe an experiment you would carry out to show that auxin causes bending of a shoot. So in this experiment, what will you do? You will paint the auxin on one side of the shoot, just like I've shown you here. So you'll take a shoot and you will paint the auxin. Auxin is a sort of a chemical which they've given you. So they've given you a young plant shoot and a sample of auxin. So then you'll paint this on one side of the shoot. Then you'll place the shoot in the dark place, and then you'll leave for about 1 to 3 days. In fact, up to 3 to 5 days. And then you will see that the shoot is going to, that side is of course going to grow more. So finally this is going to bend like this. So because it's always to the other side, in a shoot it is always on the other side on which the light is coming, but here there's no light, you just kept it in the dark, but because of the auxin it is going to elongate more on that side. So it is then you would have a similar shoot just like that, but without the auxin. This would be the control. Control means a comparison to prove that, actually it was the auxin which was causing this. And in another shoot, which we also kept in the dark, but it didn't have any auxin, that did not change shape. And then, of course, you repeat the experiment. You do it on 3 shoots with the auxin and 3 shoots without the auxin, and you keep them in the same environment. So paint the auxin on one side of the shoot, place the shoot in a dark place, leave for 3 to 5 days, use another shoot or a control which is without the auxin, and you repeat the experiment so as to prove your point. Because, in one, just by doing it once, you're not going to prove this point. So you have to be doing a number of repeats of the experiment. Then it says, explain the mechanism that result in a shoot bending towards light. So this is the shoot and light is coming from this side. Why is it going to bend towards this side? It's going to grow further towards light. Why? So auxin accumulates on this side, on the shaded side. Auxin accumulates here. Light is coming from here. So light is coming on this side. Now this is the other side. So auxin accumulates on the shaded side, and the difference in concentration on the shaded side and the light side. So cells with a higher concentration of absorb more water, and that causes unequal growth. So this side grows more, the cells elongate more, and this side grows less. So that is why you see the shoot growing towards light. So it's an unequal growth. So oxygen accumulates on the shaded side of the shoot, difference in concentration on the shaded side and light side. Cells with higher concentration of oxygen absorb more water, so this causes unequal growth. Then it says synthetic plant hormones behave in a similar way to oxygen. Describe how synthetic plant hormones are effective as weed killers. How are they effective? Large concentrations used, and the plants are stimulated to grow rapidly. Growth gets out of control, so plants die. And it's only the broad-leaved plants that are affected, which actually we want to kill, because we want to kill what are synthetic weed killers. Weed means the actual crop is not the weed. The other seeds which have germinated there, and they are taking up all the nutrients of the soil. So they're competing with the main cash crop. So maybe you have the rice crop, but in between you have little other plants growing which are no good to us. So we want to get rid of them. So that is called, those are called weed killers. Large concentrations used, plants grow rapidly, growth get out of control, and plants die. Another question on the eye. Figure 2.1 shows a section through the eye with a ray of light passing through it and four muscles labeled A, B, C, and D. Complete the table. Name of the muscle. A allows the lens to become fatter for focusing on close objects. What is that muscle? Ciliary muscle. Then iris circular muscle. What is the effect of this? Pupil becomes smaller. So that was for 2 marks. Then we come on to muscle C and D are voluntary muscles that are antagonistic. They are attached to the eye socket of the skull. Explain the terms voluntary and antagonistic. Voluntary means can be controlled by will, and antagonistic means opposing or working against each other. This is a little of the 0610 syllabus. Voluntary can be controlled by will, antagonistic working against each other, suggest the effect on the eye when muscle C contracts, eyeball is pulled to the right. Explain how the eye will return to the original position after this contraction, contraction of muscle D and relaxation of muscle C. D pulls on the eyeball, C is antagonistic to D. So this is a bit of the 0610 syllabus, not in the 5090 syllabus. Please remember that. Light passes through parts of the eye to reach the retina. Complete the flowchart by putting the following terms in the boxes to show the correct order that the light passes through them. So the first will be cornea. Then it would be the aqueous humor, cornea, aqueous humor, and then it will be the pupil. Then it will be the lens, and then it will be the vitreous humor. And then it's the retina. So cells, cornea, aqueous humor, pupil, lens, and vitreous humor. The retina contains rods and cones. Complete the table to distinguish between rods and cones. This again 0610. Type of light detected. Rods will detect shades of gray, or you can say black and white, while this is going to use color, red, green, blue. And distribution in the retina, this is spread more concentrated on the margins. more concentrated on the margins, or you can say spread over the retina, while the cones are only in the fovea. Jasmine went into a dark room from a bright corridor. Figure 4.1 has right eye before and after entering the dark room. Complete figure 4.1 by drawing the appearance of the pupil and the iris before entering the dark room, a few seconds after entering the dark room. Label the following parts of the eye on the first diagram in figure 4.1: iris, pupil, and sclera. So complete figure 4.1 by drawing the appearance of the pupil and the iris. Naturally, if from a bright corridor the person is entering into a dark room, so the pupil is going to be very small. So before entering, you can see the pupil is very small. I've drawn that in black. The green is the iris. And then of course, as it enters into the dark room, then the pupil will become larger. Label the following parts of the eye: iris, pupil, sclera. I've done that for you. Explain how the size of the pupil was changed when Jasmine went into the dark room. Simple. The pupillar muscles of the iris. And then it says explain why Jasmine could see shapes but not colors in the dark room. So role of the rods in detecting black and white images, and they are very sensitive even in low light intensities. And the cones only detect color, and cones need high intensity light to trigger them. How the size of the pupil was changed when Jasmine went into the dark room: contraction of radial muscles of the iris, relaxation of the circular muscles of the iris. Why Jasmine could see shapes but not colors? Rods detect black and white images, and rods are sensitive in low light intensity. Cones detect color, and cones need high light intensity to trigger them. This is more 0610. Please remember, this is not 5090, does not have rods and cones. This video has a mixture of questions from 5090 and 0610, and they're mostly on the coordination in the eye and the phototropism. There's one question. So I'm just posting this to help you in your just last few days of practice before the paper 2 exam.